everyone. My name is Rizal Bineswala and I am level 1 student at Rehan School Islamabad campus. जैसे होता है न्यू को कॉल माया न्यू टॉपिक आते हैं आज न्यू कॉल माया न्यू टॉपिक आते हैं रात को हमारा कॉल माया टॉपिक है द साइंस ऑफ जेनेटिक्स so this is our topic. Let's start our conversation with Maya on this topic.
Hi. Hey, I'm doing well, thanks.
Okay, Maya.
Just ready to chat. What's going on with you today?
My first question is what is genetics and why it is important in biology? Give me 100 points on it.
Got it. So, genetically, the study of genes, heredity, and variation in living organisms. It's fundamental to how traits are passed down and how life evolves. Here's 100 points covering why it's such a big deal in biology. 1. Foundation of heredity. Genetics explains how traits are passed from parents to offspring. DNA structure. The double helix of DNA discovered by Watson and Crick. 3. Gene. Definition. A gene is the basic unit of heredity. And 4. Genome composition. The complete set of genetic material in an organism. 5. Chromosomes. Structures carrying genetic information organized in the nucleus. Alleles. Different versions of the same gene. 7. Dominant alleles. Those that express their trait even with one copy present. 8. Recessive alleles. Traits only expressed with 2 copies present. 9. Genotype. The genetic makeup of an organism. 10. Phenotype. The observable characteristics of an organism. Mendelian genetics. Pioneering work by Gregor Mendel on pea plant traits. Law of aggregation. Allele pairs separate during gamete formation. 13. Law of independent assortment. Genes for different traits assort independently. 14. Squares. Tool for predicting genotype ratios in offspring. 15. Monohybrid crosses. Examining inheritance patterns of one trait. 16. Dihybrid crosses. Analyzing inheritance of 2 traits simultaneously. 17. Genetic mutations, changes in DNA sequence. 18. Point mutations, single nucleotide changes. 19. Frame, shift mutations, altering reading frame due to insertions or deletions. Chromosomal mutations, changes affect entire chromosome structure. 21. Beneficial mutations that improve an organism's fitness. 22. Harmful mutations. Mutations that reduce fitness or cause disease. 23. Neutral mutations. Mutations with no noticeable effect. 24. Genetic variation. Differences in genes within a population. 25. Gene pool. Total collection of genes in a population. 26. Evolution's mechanism. Genetics provides the basis for evolutionary change. 27. Natural selection, differential survival based on genetic traits. Adaptation, genetic changes, increasing survival and reproduction. 29. Molecular evolution, studying genetic changes over time. 30. Phylogenetic trees, reconstructing evolutionary relationships. Population genetics, studying allele frequencies in populations. Hardy, Weinberg principle, equilibrium of allele and genotype frequencies. Genetic drift, random changes in allele frequencies. 34. Bottleneck effect, drastic reduction in population size and genetic diversity. Founder effect, establishment of new populations with limited genetic variation. Gene flow, transfer of genes between populations. 37. Non-random mating, influences genotype frequencies. 38. Inbreeding depression, reduce fitness from mating with close relatives. Quantitative traits, traits influenced by multiple genes. 40. Polygenic inheritance, inheritance involving multiple genes. 41. Epistasis, interaction between genes affecting phenotype. 42. Environmental influence, how environment affects gene expression. 43. Norm of reaction, range of phenotypes for a genotype in different environments. 44. Genetic engineering, direct manipulation of genes. 45. Recombinant DNA technology, combining DNA from different sources. Gene cloning, creating multiple copies of a 47. Polymerase chain reaction, amplifying specific DNA segments. 48. Gene therapy, introducing genes to treat diseases. 49. GMOs, genetically modified organisms. Agricultural applications, developing pest-resistant crops. 51. Medical diagnostics, genetic testing for disease prediction. 52. Personalized medicine, tailoring treatments based on genetic makeup. 53. Printing for identification. 54. Ethical considerations, debates surrounding genetic technologies. 55. Genetic counseling, providing guidance on genetic risks. 56. Genome sequencing, determining the order of DNA bases. 57. Human Genome Project, mapping the human genome. 58. Comparative genomics, comparing genomes of different species. 59. Functional genomics, studying gene functions. Proteomics, studying protein production and function. Transcriptomics, studying RNA expression patterns. C2. Metabolomics, analyzing small molecules involved in metabolism. 63. Epigenetics, changes in gene expression without DNA sequence changes. 64. Histone modification, altering histone structure to regulate gene expression. DNA methylation, adding methyl groups to de-silence genes. RNA interference, using RNA to silence gene expression. CSNOT, CRISPR, Cas9 technology, gene editing tool for precise DNA modifications. Editing, making targeted changes in genomes. 69. Synthetic biology, designing and constructing new biological parts. 70. Systems biology, studying interactions between genes and environment. 1. Regulatory networks, complex interactions controlling gene expression. 72. Developmental genetics, studying gene roles in development. 73. Homeobox genes, master regulatory genes controlling body plan. 74. Cancer genetics, understanding genetic basis of cancer. 75. Oncogenes, genes promoting cancer development. 6. Tumor suppressor, genes inhibiting cancer development. 77. Genetic predisposition, risks for certain diseases. 78. Immunogenetics, studying immune genetics. Pharmacogenomics, studying how genes affect drug response. Genetic neurogenetics, study genetics of neurological disorders. 81. Behavioral genetics, study genetic influences on behavior. 82. Twin studies comparing identical and fraternal twins. 83. Adoption studies comparing adopted children to biological parents. 84. Population structure, how genes vary in different human populations. 85. Ancient DNA, studying DNA of extinct organisms. 86. Forensic genetics, applications of genetics in criminal investigations. 87. Gene drive technology, genomes through inheritance. 88. Conservation genetics, applying genetics to conservation efforts. 89. Evolutionary biology, studying how development evolves. 90. Epigenetic inheritance, passing epigenetic changes to offspring. Microbiome. 91. Microbiome genetics, study genetics of microorganisms. Genetically modified foods, safety, debates on the safety of GM foods. 23. Intellectual patenting of genes and genetic technologies. 24. Bioinformatics, applying computational tools to analyze genetic data. Biostatistics, analyzing genetic data using LC research, examining societal impact. Germline editing, genes in eggs or sperm. 98. Somatic cell editing, editing genes in non-active cells. Personalized nutrition, tailoring diet based on genetic dispositions. 100,000. The future of genetic, ongoing research and emerging technologies. That's pretty comprehensive view of why genetics is so important. It's woven into so much of what we know about life. What got you interested in this specifically?
Okay, my next question is for you. Who is known as the father of genetics and why? Give me 100 point on it.
I'm having trouble catching some of those words clearly. You were asking about the father of genetics, is that right? And 100 points on who that is and why?
Yes, Maya.
Okay, so when we the father of genetics, working about Gregor Mendel, he's the one who really laid the foundation for how we understand. Here are 100 points about him. 1. Austrian monk. Mendel was an Austrian monk living in a monastery. Monastery garden. He could famous in the monastery garden. 3. Pea plants. He used pea plants for his experiments. They were perfect for observation. 4. Experimental design. Mendel meticulously planned his experiments with 5. Traits. He focused on traits like flower color, pea shape, and height. 6. True breeding lines. He started with plant true breeding for specific traits. 7. Controlled pollination. He carefully controlled pollination between 8 generations observed. He tracked traits across multiple generations. 9. Quantitative approach. He used math to analyze his results. This was unusual for the time. 10. First generation. In the first generation, traits did blend but stayed distinct. 11. Law segregation. Each individual carries 2 alleles for each trait that separate gamete formation. 12. Independent assortment. Genes for different traits assort independently. Domain dominance. Some traits matters in hybrids. 14. Phenotype ratios, meta-specific ratios in his results, 3:1. 9, 3, 3:1. 15. First scientific theory. Developed what we recognize today as the first scientific theory of heredity. 16. Manuscript publication. Published his work in 1866, but it was largely ignored at the time. 17. Rediscovery. His work was rediscovered in 1900 by Hugo Dives, Karl Correns, and Erich von Chermach. 18. Recognition. After discovery, he finally gained recognition for his contributions. Mendel's legacy. He is now universally hailed as the father of genetics. 20. Basic principles. Establish the basic principles that govern inheritance. 21. No understanding of DNA. He didn't know about DNAs or chromosomes. He worked at a more observable level. Mathematical approach. His use of mathematics revolutionized biology. 23. Rejection of inheritance. He disproved the previous idea that traits blend in offspring. 24. Modern genetics. Foundations. His work provides the foundation for modern genetics. 25. Understanding disease inheritance. His principles help understand hereditary diseases. 26. Crop improvement. Genetics principles assist in improving crop varieties. 27. evolutionary biology. Genetics helps study the mechanisms of evolution. 28. Modern agriculture. Genetics assists with selective breeding practices. 29. Plant breeding techniques. Mendel's principles influence plant breeding programs. 30. Animal husbandry. Mendel's principles assist animal breeding. On molecular biology link, his work provided a conceptual link to molecular biology. 32. Gene identification led to the later identification. 33. Genome mapping helps in genome mapping studies. 34. Gene sequencing enabled gene sequencing efforts. 35. Biotechnology applications forms the basis of biotechnology applications. Personalized medicine. Genetics enables personalized medicine approaches. Counseling provides a foundation for genetic counseling. 38. Prenatal genetic testing allows for prenatal genetic testing. 39. Forensic genetics principles used in forensic analysis. DNA fingerprinting. Mendel's legacy in DNA fingerprinting. 41. Species conservation utilized in species conservation genetics. 42. Population genetics studies applicable to population genetics research. 43. Quantitative trait loci influences study of quantitative traits. Evolutionary biology advances in evolutionary biology understanding. 45. Genetics education fundamental to genetics education. Biological research basis for biological research across disciplines. Scientific method importance. His work highlights the importance of scientific method. Experiment control demonstrates the importance of controlled experiments. 49. Observation skill. He had keen powers of observation. Logical reasoning displayed excellent logical reasoning. Analysis capabilities excelled at data analysis. Pattern recognition could recognize pattern in data. 53. Documentation kept meticulous records. Hypothesis formula ability to formulate testable hypotheses. 55. Theory building created and tested his theories. 56. Paradigm shift shifted the understanding of heredity. 57. Reduction biology approach emphasizing reductionist principles. 58. Scientific revolution contributed to the scientific revolution. 59. Impact on agriculture profound impact on agricultural practices. Impact on medicine transform care through genetics. Biotechnology revolution foundation of biotechnology revolution. Personalized disease treatment revolutionized disease treatment options. 63. Future genetic technologies inspires development of new technologies. 64. Under complex traits assists research of complex traits. Gene therapy development guides the development of gene therapy. 66. Genome editing contribute genome editing research. 67. Precision breeding enables precision breeding. 68. Disease resistance breeding improves crop disease resistance. 69. Yield improvement enhances crop and livestock yields. Nutritional enhancement allows nutritional enhancement in agriculture. 71. Adaptation research understanding of adaptation mechanisms. 72. Behavior genetics basis for studying genetic influence on behavior. Brain development provides insight development genetics. 74. Neurological disorders aids neurological research. 75. Cancer studies enhances cancer genetics research. 76. Immune function contributes to immune function studies. Metabolic disease research impacts metabolic disease research. 78. Aging research influences studies on genetic aspects of aging. 79. Population diversity aids diversity in genetics. Human evolution understanding enhances human evolution knowledge. 81. Paleogenetic analysis guides studies on ancient DNA analysis. 82. Phylogenetic reconstruction contributes to phylogenetic reconstruction. 83. Bioinformatics integration advances bioinformatics advancements. 84. Biostatistical analysis enables biostatistical method applications. 85. Ethical framework provides basis for ethical framework development. 86. Data management highlights the importance of managing data. 87. Laboratory equipment advances supports laboratory equipment development. 88. Research funding promotes research funding through discoveries. 89. Collaboration networks creates collaborative research networks. Innovation catalyzer serves as catalyst for genetic innovation. 91. International collaboration takes international genetic research. 92. Nomination enables genetics knowledge dissemination. Scientific literacy and scientific understanding for public education. Educational curriculum forms foundation for educational curriculum. Communication skills highlights effective science communication. 96. Open access data provides resources for open access data projects. Research career inspiration inspires future genetic research careers. 98. Mentoring support supports mentoring to develop new talent. Innovation support contributes to fostering innovation across biotech. Winded. Long-lasting impact continues impact across multiple scientific disciplines. You still there, Ali Saleh?
Yes, Maya. My next question is for you. What is DNA and why and what role does it play in heritage? Give me 30 points on it.
Right, so DNA, deoxyribonucleic acid, is the molecule that carries the genetic instructions for all living organisms. Here, he points about what it is and its role in heredity.
So this is our conversation, Maya. I hope you like it. See you next time. Allah Hafiz. Pakistan Zindabad. Rehan Zindabad. Allah Hafiz.